Today we'll be discussing how eukaryotic genome replication and expression differs from bacteria. So if you haven't watched the video yet on bacterial replication and expression, that's a great place to start. So why do we care about differences between eukarya and bacteria when it comes to gene replication and expression? Well, for one, it allows for some really cool technology. This tobacco plant might have bacterial genes in it. Tobacco is super well studied, and it's a common organism that's used for genome modification and engineering. Bacterial genes are actually inserted into the DNA of the tobacco plant. And keep in mind, tobacco is a plant, and therefore it's a eukaryotic organism. So we're actually mixing DNA from two different domains of life, which is pretty incredible. Transgenic tobacco can be engineered to produce useful proteins or enzymes. It can also even make sweeteners like mannitol, for an example. Or they can be engineered to contain some bacterial genes that help the plant resist pathogens and endure stress. Or did you know that the insulin many people with diabetes use doesn't come from animals anymore? It's actually made by bacteria. Scientists take the human gene for insulin and insert it into a bacterial plasmid, which is then placed inside E. coli cells. Even though bacteria don't naturally make insulin, they can still read the human gene, transcribe it into RNA, and translate it into protein using their own machinery. With the right signals added to the gene, the bacteria produce insulin that's chemically identical to the human version, allowing us to mass produce life-saving medicine in a simple microbe. But there are major differences between human gene expression and bacterial gene expression. So these scientists had to use knowledge of both pathways and some ingenuity to be able to make this system work. So by the end of this video, we'll be able to understand just how they were able to pull this off. Let's start with the big picture. Bacterial and eukaryotic genomes are organized very differently. Bacteria usually have a single circular chromosome, relatively small in size and generally lacking extensive packaging proteins. They do have DNA binding proteins, but not the complex histone-based chromatin found in eukaryotes. Eukaryotes, by contrast, then have multiple linear chromosomes, each much larger than a bacterial chromosome, generally speaking. These are tightly packaged around histones into chromatin, which adds an additional layer of regulation. In bacteria, there's typically a single origin of replication per chromosome. But eukaryotic chromosomes are much larger and linear, so they require many origins of replication spread across each chromosome. That brings us to the origin recognition complex, or the ORC. This is a protein complex unique to eukaryotes that binds origins of replication and recruits the rest of the replication machinery. Replication enzymes also differ. In bacteria, DNA polymerase III is the main replicative enzyme. In eukaryotes, DNA polymerase delta and epsilon carry out leading and lagging strand synthesis. Interestingly, these polymerases are also in the same family as the polymerases that are found in some archaea for genome replication. Eukarya can face an extra challenge due to their linear chromosomes. First, let's recap the general steps of replication so that I can show you where the obstacle lies. And by the way, I'm going to keep it super simple here. So step 1, helicase opens up the DNA, making a replication bubble. Step 2 would be that we're laying down our RNA primers. Step 3, thanks to DNA polymerase, we're going to lay down some nucleotides. Step 4, we have our Okazaki fragments. Step 5, we remove the primers, then DNA polymerase fills in the gaps. And here's where the issue comes into play. DNA polymerase needs a 3' hydroxyl end to latch onto to work. So every time replication reaches the ends of the chromosome, the lagging strand can't be fully copied, leading to progressive shortening. To fix this, eukaryotes use telomerase. Telomeres are made up of repeated DNA sequences, and these repeats match up with the RNA template that's built right into the telomerase enzyme. Telomerase uses that RNA piece to base pair with the very end of the single-stranded part of the telomere. Once it's paired, telomerase acts like a reverse transcriptase. It reads the RNA template and adds new DNA to the single-stranded end, making it longer. When the single strand has been extended enough, primase can lay down a short RNA primer, and then DNA polymerase comes in and fills the gaps with regular DNA. Now let's shift to gene expression. In transcription, bacteria have a single RNA polymerase, while eukaryotes have 3. RNA polymerase I makes ribosomal RNA. RNA polymerase II makes messenger RNA, or mRNA. And RNA polymerase III makes tRNA, or transfer RNA, and 5S ribosomal RNA. Sigma factors are usually single proteins that temporarily attach to bacterial RNA polymerase and guide it towards the promoter. Once transcription starts, they usually fall off. Eukaryotes don't use sigma factors. Instead, they rely on a suite of general transcription factors. Eukaryotic mRNAs also undergo extensive processing. The primary transcript, or pre-mRNA, must be capped, polyadenylated, and spliced before it becomes a mature mRNA. One powerful feature here is alternative splicing, where the same gene can give rise to multiple protein products depending on which exons are included or excluded. Finally, translation initiation also differs. In bacteria and archaea, the ribosome recognizes a Shine-Dalgarno sequence just upstream of the start codon. In eukaryotes, initiation is distinct. The small ribosomal subunit binds to the 5' cap of the mRNA and scans along until it finds the first AUG start codon in the right context. So here's the big takeaway. One reason we can program bacterial cells to uptake human insulin and produce it is because we understand both similarities and differences between gene expression in both of these different domains of life. So we can't just take the insulin gene straight from a human cell and put it into bacteria. Why? Because eukaryotic cells have certain processing steps that bacterial cells just simply don't. For one, human genes contain introns, and pre-mRNA has to be processed by splicing to take the introns out. Bacteria just don't have the machinery to do that sort of processing. The workaround is actually pretty clever. So scientists will take the already processed mRNA for the human insulin gene and then use an enzyme called reverse transcriptase to turn it back into DNA. And then we can take that DNA, put it into a plasmid, and get that plasmid into a bacteria. And at that point, when the bacteria goes ahead to transcribe and translate that gene, it doesn't have to do any of the processing steps that we usually need. In other words, by understanding where bacteria and eukarya overlap and also how we're different, we can then accomplish really amazing things like turning bacteria into insulin drug factories.